Hello friends, welcome to my channel. Now in today class we will going to learn about monoclonal antibodies. Okay, so we will learn the production of monoclonal antibody, how the monoclonal antibodies are produced. First we need to understand the meaning of monoclonal. Okay, mono means one, right? Let me change the thickness. Mono means one, clonal means copy, right? Clone means copy, right? So the monoclonal means copy of one antibody. Antibodies, we know antibodies fight against the antigen, right? So this is the meaning of monoclonal antibody. Mono means one, clone means copy. Now how we produce monoclonal antibodies? So first what we do, we inject the antigen into the mouse. Okay, so the mouse is vaccinated to start the formation of antibody. Now how the mouse will start the formation of antibody? By injecting the antigen. So first we inject the antigen into the mouse body. Okay, antigen. And we inject, when we inject the antigen, the mouse body start producing the antibody to fight against that antigen that we have injected into the mouse. Now it is the spleen cell into the mouse that help to produce the antibody. Okay, so which part of the mouse produce the antibody? It is the spleen cell. So what we do, we extract that spleen cell from the mouse, because it is the spleen cell that produce the antibody. So let's remove that spleen cell. So the spleen cell that form the antibodies are collected from the mouse in an operation. So we do a very minor operation on a mouse and we remove the spleen cell that produce the antibody. Now what we do, we fuse this spleen cell with the myeloma cell or the tumor cell. Okay, now what is a cancer cell or a tumor cell? What is the important, or what is the specialization of that cell? They keep on reproducing themselves, they keep on dividing themselves, right? That is the nature of the myeloma cell or a cancer cell or a tumor cell, which is a myeloma cell, because it is keep on dividing itself. So what we do, we fuse this spleen cell with the myeloma cell. Okay, and this form a hybridoma cell. Hybridoma means the combination of two, right? So you have added these two cell and it form a hybridoma cell, and these are grown in a lab and produce a copies of antibodies. Because see, now how it produce the copies of antibody, because this is the nature of the tumor cell to keep on dividing, right? How the cancer form? Cancer is basically the uncontrolled division of the cell. So when this uncontrolled division of the cell takes place, it also divide the antibodies. Okay, so that is how it keep on producing the n number of antibodies. And by in the lab we extract the antibodies from the tumor cell. That is how we produce the, or production of the monoclonal antibody take place. Let me explain you again. Okay, so first we inject the antigen into the mouse. Mouse produce the antibodies. We extract the spleen cell from the mouse, hybridize or do the fusion with the tumor cell or the myeloma cell, and this form a hybridoma cell. And we grow this tumor cell into a lab and they start producing the antibodies, and then we separate the antibodies, and that is how we produce the monoclonal antibody. Now what are the uses of this? Why we are using the monoclonal antibody? So these are the uses. So we use monoclonal antibody in the pregnancy test kit. Okay. See, the pregnant women release hormone, that is HCG hormone, in their urine. So when they drop the urine into the pregnancy test kit, that get bind to the, yes, it bind to the antigen, and that is how the lady came to know about that she is pregnant. So the monoclonal antibody used for the pregnancy test, and we also use for the cancer diagnosis and the treatment, and we also use the monoclonal antibody for the diagnose to diagnose and find the infection of HIV, AIDS, chlamydia, herpes. So these all are the uses of the monoclonal antibody. Okay, so what they do in the cancer, they bind with the cancer cell, clump together, okay, clump together the cancer cell, so it will be easy to detect the cancer cell and remove that cancer cell. That is how the treatment and the diagnosis of the cancer cell done with the monoclonal antibody. Okay, uh, I hope this part is clear, and this is a very short topic and very easy also, right? So please like, share, and subscribe to my channel. See you in the next class. Bye.